Hello, everyone. Nice to see all of you for our first UCI Biosci Virtual Dean's Distinguished Lecture. Before I introduce today's lecture and our guest speaker, I just want to thank everyone in the Biosci Dean's Office for helping put this virtual lecture together and for making the event a success. As many of you know, this lecture was born out of a desire for us to better inform our local community here in Orange County and Southern California about all the cutting edge research being done in the School of Biological Sciences at UC Irvine. Obviously, because of the virtual nature of this, we're able to even expand beyond our traditional borders. As many of you know, in the past, these distinguished lectures have been hosted in a very large venue on campus. And while I regret that we can't be on the campus right now, we wanted to continue the series. And after all, what better time to shine a light on life science research during an unprecedented global pandemic? As many of you know, as biologists, we have been fervently studying many aspects of this novel coronavirus disease. And in fact, our school's organizing theme of Mind, Body, World continues to position us to make progress against COVID-19 and future pandemics. The importance, I think, of supporting life science research and education has never been more compelling than it is right now. We appreciate all of you joining us on Zoom today to learn more about the effects of coronavirus on the central nervous system. As a matter of fact, I can't think of a more timely topic. And just as I was about to come on, I got a little notification from CNN that just said that recently California became the second state in America to surpass over 1 million cases of COVID-19. And so with that, I think we're very lucky to have our own speaker, Professor Thomas Lane, who joined the UCI School of Biological Sciences faculty back in 1998 and is now a Chancellor's Professor of Neurobiology and Behavior. He has a true expert on coronavirus infection and has a very prolific and a very detailed research career studying the effect of coronavirus on the brain for over 2 decades. He is a well-regarded scientist. He is eagerly sought after to be a collaborator by many of us on campus and throughout the nation. And he has played many roles at UCI, including serving as director of the Multiple Sclerosis Research Center and associate director of the Institute for Immunology. He's been awarded a National MS Society Collaborative Center Award and a CIRM Early Translation Award. And today, Tom will share with us his research about coronavirus, about learning how SARS-CoV-2, the virus that causes COVID-19, gets into the brain, and what his team has discovered for its impact on conditions like diabetes and cardiovascular disease. So let's all please welcome Professor Tom Lane.
Okay, let's see. I think everybody can hear me. Frank, thank you so much for that extremely kind introduction, as well as the opportunity to talk about something that's clearly very important to us, as well as a number of people, and this is examining the effects of COVID-19 on the central nervous system. I would also like to extend my sincere thanks to everyone that's been able to attend the lecture today. I know everybody's very busy, so thanks for taking the time and coming out to hear what we and others on the UCI campus are doing. All right, so what I'd like to do is provide an outline of the talk this afternoon, and I'm going to begin by providing an introduction to coronaviruses. Then I'll move into SARS-CoV-2, which is the etiologic or causative agent of COVID-19. discuss some of aspects related to the virology and immunology of this disease. Then move into CNS conditions or symptoms that occur in some individuals following SARS-CoV-2 infection. And then finally, I'd like to conclude, as Frank pointed out, on some of the preclinical mouse studies that we've been doing using SARS-CoV-2 infection of susceptible mice, and specifically looking at the CNS disease that occurs in these animals. And then finally, I'd like to conclude with some perspectives, and then I'd be happy to answer some questions from the audience. Okay, so coronaviruses, as this title implies, this is a virus that we've been living with for decades, if not longer. And here are 4 transmission electron micrographs depicting SARS-CoV-1, SARS-CoV-2, MERS-CoV, and then mouse hepatitis virus, which is a mouse coronavirus. And the point of me putting these images up here is that although these are different types of coronaviruses, you can really see from the images depicted that how these viruses acquire their name. They have these solar crowns or coronas. And this is the result of the spike glycoprotein shown here in this cartoon, which extends furthest from the surface of the virion. And I would imagine most of you have probably heard of the spike glycoprotein, because when social media is discussing the efficacy of vaccine or vaccine development, most are talking about directing immune responses that target the spike glycoprotein, because this mediates attachment to the host cell receptor. And I'll talk about that in a little more detail later. In addition to the spike glycoprotein, there are additional structural proteins that are very important. You have the membrane protein, which is shown here, as well as the envelope protein. And these proteins all work together to encircle or protect the RNA genome, which is shown here by this large black line. And the coronaviruses are single-stranded RNA viruses. In the virology world, you have DNA viruses and RNA viruses, and then retroviruses, but coronaviruses are RNA viruses, and they are among the largest known, not just in physical size, but really what this relates to is the size of the RNA genome. So these are roughly 31 kilobases, or 31,000 base pairs in length. And the 4 coronaviruses I'm showing here are a member of the beta coronavirus family. There are actually 4 families of coronaviruses. Alpha and beta coronaviruses are primarily able to infect and replicate in mammals. And then the gamma and delta coronaviruses are primarily restricted to either fish or birds. And so for the beta coronaviruses, because they have a wide range of animal hosts, which include humans, obviously, but also pigs, cattle, rodents such as mice and rats, bats, and camels, they have a variety of different diseases that can be initiated in these animals upon infection. And the symptoms can be varied, and they can be either benign, that is really no evidence of infection at all, or they can be much more severe, which can include pneumonia, diarrhea, peritonitis, which is inflammation of the peritoneal cavity or the gut, as well as neurologic disease. And this is primarily characterized by encephalitis, which is an inflammation of the brain. And this can be accompanied by neurodegeneration. So just very quickly, there are a number of neurotropic animal coronaviruses. That means coronaviruses that are able to infect and replicate within the central nervous system. And one of these is murine coronavirus. And as Frank alluded to, for the past 2 decades, our laboratory has really devoted a lot of attention in understanding the immune mechanisms that are elicited in response to infection of the central nervous system of susceptible mice by these murine coronaviruses that either help control the virus or contain it, as well as potentially contribute to neurodegenerative disease. In addition, we've explored for quite some time molecular mechanisms that govern neuroinflammation in response to infection of the CNS. If you own a cat, you've probably heard of feline infectious peritonitis virus. This is a very serious disease in cats. it does induce this inflammation of the gut, and it is under many circumstances lethal, but the virus itself is CNS penetrant. It can get into the central nervous system of these cats and also cause problems. And then finally, another example I've included is the porcine encephalomyelitis virus. And so this is a real problem of the swine industry, where these viruses can get into the CNS and cause or just wreak havoc for farmers that build pig herds. All right, but the coronaviruses that you're probably most familiar with, either directly or indirectly, are these 4 human coronaviruses. And these are really associated with mild seasonal respiratory illnesses. And so there are 4 of these, as I indicated, 229E and OC43. These were discovered in the 1960s at the University of Chicago, as well as the NIH. And then after the first SARS-CoV-1, if you will, infection that occurred in 2002, this resurrected a lot of interest in coronavirus research. And then 2 additional coronaviruses were discovered in Hong Kong and then the Netherlands. And so these collectively are referred to as the common cold coronaviruses, or CCC. And roughly about 15% of upper respiratory tract infections are associated with these 4 coronaviruses. They, from a clinical standpoint, they're almost indistinguishable from rhinoviruses, which are another quote unquote common cold. And what's interesting though, about 90% of people tested have antibodies to at least one of these particular viruses. So we've all probably been exposed. But what I want to transition to and use as a springboard into the rest of the talk are the coronaviruses you've definitely heard about. And so I'll begin with this severe acute respiratory syndrome virus, or SARS, which is now known as SARS-CoV-1 or SARS-1. And so this emerged in 2002. And late 2002, the first known cases of an atypical pneumonia were described in Fushan, China. By February of 2003, China had reported over 300 cases of these acute respiratory syndromes occurring in the Guangdong province. By April of that year, it was identified as a coronavirus as the causative agent. And then it spread to different countries around the world, but ultimately it was contained. In terms of how this got into humans, it's clear now that bats were the original host of the virus. But then at some point it was able to spread into civet cats as well as raccoon dogs. And so these are animals that are often found in these exotic wet markets at various locations throughout China. And it was during these wet markets potentially that it jumped into humans. All right, and that in itself is not that big of a deal, because a lot of viruses jump from animals into human. But this is a respiratory virus, and the main thing that's very important to keep in mind, once it jumped into humans, it was then able to spread human to human through aerosolization of respiratory droplets containing the virus. Importantly, no antiviral therapies are effective against this SARS-1, and there are no vaccines to date. And I found this cover of Time magazine, and note that it's May 5th, 2003. But so many things are relevant in this cover to what we're dealing with right now with SARS-2. We're all wearing masks. We're curious about mechanisms by which the virus spread, and then how scared should you be? All right, and so we've all asked these questions, and certainly we're all wearing masks every day. Here is just a map that I compiled from the WHO that represents the cumulative cases from 2002 to 2003 for SARS. And you can see that it was heavily concentrated in China, but it did spread, usually by air travel, to other countries around the world and continents, including North America, with Canada having slightly more cases than the US. But by near the end of 2003, WHO indicated this virus was contained, all right? And so one reason why it was, even though there weren't any antivirals or vaccines, is because the virus is very hard to transmit. You had to be very sick in order to spread the virus. And the primary people that were affected by the virus were either family members that lived in close proximity with an infected individual or healthcare workers. All right, and then so once that was discovered, you could quarantine or isolate infected patients, and that really dampened the spread of the virus to where now we really have no problems with this. So now let's jump to 2012. And this is when the Middle Eastern respiratory syndrome, or MERS coronavirus, emerged. First reported cases appeared in Saudi Arabia in 2012, or September of 2012. And this virus was primarily confined to countries either in or near the Arabian Peninsula. And then in November of 2012, the virus that was isolated from these people was identified as a novel coronavirus. This was a paper that appeared in the New England Journal of Medicine. And similar to SARS-1, bats are the original host. However, in this scenario, it spread to camels, and then camels are the vector to humans. And again, so once humans acquired it, they were able to efficiently spread it from human to human. Similar to SARS-1, there are no antiviruses or antiviral therapies available or vaccines. I acquired this image from the NIH website. I like this because this depicts MERS coronavirus particles colored in purple. They're not really purple, that have been infecting a cell monolayer in the laboratory. These are called Vero E6 cells. But it really kind of shows how the virus can spread from cell to cell. And as I indicated, this virus, there weren't that many cases, about 2,500 cases, yet it had close to 900 deaths. And so this is a case fatality rate of 37%. And I forgot to mention this, for SARS-1, the mortality rate was around 10%. But here again, as you can see, the virus is confined almost exclusively to Saudi Arabia, but also in areas surrounding this. Other countries did get involved, but it was very minor hit. Again, why? Because the virus doesn't spread very efficiently. You have to be very sick with symptoms in order to spread the virus. Family members, again, and healthcare workers were the primary patients or people that were affected by the virus. And I just want to put one thing in perspective. If you consider the entire number of cases that we just discussed with SARS-1, and which is, I think there were 8,000 cases total and 2,500 cases here, that's almost a daily infection rate, or a fraction of the daily infection rate in the United States alone for COVID-19, which now allows me to bring up COVID-19. So as I indicated, the causative agent is another coronavirus called SARS-CoV-2. It causes the disease coronavirus disease 2019, or COVID-19. And I put this article up because I think it really captures what's very important. And the title is Another Decade, Another Coronavirus. And I think that's very important because we had SARS-1 in the early 2000s, MERS-CoV 2012, and we still are dealing with this virus right now. It's not been eliminated. And then SARS-CoV-2. This article was written by Stanley Perlman, who is an outstanding clinician scientist at the University of Iowa, who is a internationally recognized expert in coronavirus pathogenesis. So just a quick background on this. I captured this image because I actually remember reading this article in Science, which is a very high profile scientific journal. And note the timestamp, January 3rd, 2020. And what caught my eye was novel human virus. Pneumonia cases linked to seafood market in China cause concern. And so I immediately thought, well, this sounds a lot like SARS-1. And indeed, if you look at the time frame here, which I'm sure everyone is familiar with because we all lived through it, we had December 31st, 2019, the WHO is informed of pneumonia cases of unknown cause in China. By January 7th of 2020, this identified cause of pneumonia was a novel coronavirus. 2 days later, on the January 9th, first death was reported in Wuhan, China. By February 11th of 2020, the virus was named SARS-CoV-2, and the WHO names the disease COVID-19, or coronavirus disease 2019. By March 11th, 2020, it's declared a pandemic status by the WHO. This is just an image, or images I captured from Johns Hopkins public coronavirus, or COVID-19 website, showing the distribution of cases around the world. This is the United States, showing the distribution of cases. And then finally, I thought this was a great image captured from the San Francisco Chronicle at the beginning of the pandemic, stay at home, which is, of course, something we all did. Now, the one thing that SARS-1, MERS-CoV, and SARS-2 have in common is they are primarily, again, a respiratory disease in which the lung is involved. And in severe cases, it's people will develop an interstitial pneumonia and die from that. All right, so that's what they have in common. I'll go over some other aspects that set SARS-CoV-2 apart from the other two coronaviruses. But first, I just wanted to go over very quickly the immune response to SARS-CoV-2 infection, because a lot of us have been hearing about different aspects of the immune response, antibody responses, they come up, they go away. And this is also very important with regards to the development of effective and safe vaccines. And while I'm concentrating on SARS-CoV-2 and the immune response that ensues, this actually is very similar to many different types of viruses, okay? So here we have infection by SARS-CoV-2. It goes into the lungs. And what happens very quickly, within primarily days, you have the virus is taken up by a variety of different cell types called antigen-presenting cells. And here is a dendritic cell, or DC. And the job of these cells is to engulf or capture the virus. And then what it will do is two things. It will migrate back to lymphatic tissue or lymph nodes. And so if you're sick, you know, you have swollen lymph nodes and they get inflamed. That's because your immune system is developing a response to that infectious agent. So these antigen presenting cells like DCs go back to the lymph nodes. And what they do is they present antigens or viral proteins or peptides to components of the adaptive immune response, okay? So these APCs, or antigen presenting cells, are components of the innate immune response, and their job is to communicate with cells of the adaptive immune response, and specifically T cells and then subsequently B cells. So very quickly, they're just different types of T cells, some that elaborate or secrete a lot of cytokines that allow for cell-to-cell communication. Other T cells help regulate or dampen inflammation. Other T cells that are elicited are cytotoxic T cells, or CTLs. What the job of these cells, the exclusive job almost, is to kill virally infected cells. And then you have another T cell subset called follicular helper T cells. And what these cells do is they provide information to B cells that have this antigen receptor on its surface. These cells expand and can secrete antibodies that are specific for viral proteins. And in this cartoon, we're showing that antibodies can be specific for the spike glycoprotein shown here. And why that is important is because if you have antibodies that can bind to the spike glycoprotein, it can inhibit the ability of that virus to infect a cell and subsequently spread, okay? Now, this is part of the acute immune response. And upon resolution of infection, many of these lymphocytes will go away, but you will have two remaining populations of T cells and antibody secreting cells, or B cells. And these are called memory T cells or B cells. And why are these important? Because if you become re-exposed to a virus that you've generated immune response to, these memory T cells are rapidly expand and migrate to the site of infection to very rapidly eliminate the invading viral pathogen, probably before you become symptomatic. And so the question that really is important with regards to SARS-CoV-2 is, A, are we generating sufficient populations of memory lymphocytes so that if you have survived or gotten through COVID-19, if you are re-exposed to SARS-CoV-2, are you protected? Are these memory lymphocytes sufficient to protect you from reinfection? And this is extremely important, all right? In addition, this whole formation of memory cells is the basis of vaccination. And so what people all around the world, companies around the world, are trying to elicit or build vaccines that generate effective memory populations of cells to protect us from subsequent reinfection or just infection primarily. All right. Now, clinical features of COVID-19, I'm sure we're all familiar with this, but age is a major risk factor. In general, if you are relatively young, as defined as less than 50 years of age, which technically I'm not young anymore, you are either asymptomatic or will have very mild disease. Mild disease meaning either fever, fatigue, or a dry cough. You might get a mild pneumonia, but it'll resolve. Now, as you get older and or have comorbidities, which means like a pre-existing heart condition or metabolic diseases, you have an increased likelihood of developing more severe conditions, including shortness of breath, and in severe cases, you might have to be admitted to the hospital and into an intensive care unit. Close to 70 years old or greater with multiple comorbidities, so again, obesity, diabetes, heart conditions, then you have a much greater chance of having severe disease, being hospitalized, developing what's known as ARDS, or acute respiratory disease distress syndrome, which is a massive inflammatory response in the lungs. That's a result of overt cytokine expression. You probably heard the term cytokine storm. Well, that mediates this inflammatory response in the lung, and this can be fatal in certain people. In addition, cardiac injury can occur in elderly individuals, as well as multi-organ failure. All right, now, what is the receptor for SARS-CoV-2? Because this is important in understanding the biology of the virus. So all viruses need to get into a host cell in order to replicate. They need to get into the cell, release their genome, and then pirate or employ host cell machinery in order to replicate and make progeny virus. No different for SARS-CoV-2. It utilizes a receptor that's known as angiotensin converting enzyme 2, or ACE2. And so how you have to think about this is basically, if you needed to get into a locked house, you have a key and you insert it into a lock, open it up, and you go in. It's no different for a virus getting into a cell. The spike glycoprotein is the key. The lock is ACE2. This engagement of these two allows then subsequent entry of the SARS-CoV-2 virus into the host cell, and then the viral genome is deposited. There is a hierarchy in terms of expression of ACE2 protein and mRNA in different tissues throughout the body, and this to some extent will dictate the ability of the virus to spread. Oh, one other thing, ACE2 under normal circumstances regulates normal vascular integrity and function. And in fact, there are drugs available right now that target ACE2 for people that have problems with hypertension. All right, so what does SARS-CoV-2 or COVID infection of the lungs look like? These images I'm showing here are the result of a wonderful collaboration I've had for years with a truly outstanding clinician scientist, Dr. Rob Edwards, who is a professor in the Department of Pathology here at UCI. He has been able to perform autopsies on individuals that passed away from COVID-19. This down here is lung tissue acquired from a 60-year-old male that had extensive comorbidities. And what I want to first focus your attention to are normal lungs. So this is just normal lung tissue that was acquired from an individual years ago that we cut to look what an uninfected lung looks like. And really what I want you to look at under low power as well as high power is that overall there's a reduced cellularity and there's a lot of air, right? And that's what you want your lung to look like, right? Because the lung is for gas exchange. In contrast, if you look under low magnification of this individual that passed away from COVID-19, you see not a lot of air, but an enormous amount of inflammatory cells that have migrated into the lung. And then the brown staining here is viral RNA. And all of this work was done by, the staining and everything was done by an outstanding second-year graduate student, Hema Oliveria, that's in my laboratory that she's just really a workhorse. And she did all the staining here. And what you can appreciate is this massive involvement of a number of cells that are infected by virus and that ultimately led to the passing of this individual. Okay, so what are some of the unusual clinical manifestations of COVID-19 that sets it apart from SARS-1 and MERS-CoV? One is there is a heart disease that's been reported in individuals as well as kidney damage. There is also inflammation of blood vessels. You've probably heard quite a bit about stroke, and this is the result of thrombosis or coagulation that can occur. There also are hyperinflammatory syndromes that can occur in both adults as well as young children. And then a very important aspect that's related to SARS-CoV-2 or COVID-19 is that you can have asymptomatic spreaders, all right? And what that means is that you can acquire the virus, you do not have any symptoms that would make you aware that you have been infected. So you don't have a fever, a runny nose, et cetera, yet you can shed or spread the virus through respiratory droplets, all right, which is the primary means of spread, okay? And that's in stark contrast to SARS-1 and MERS-CoV, where you had to be very symptomatic in order to spread the virus. In addition, there's an increased frequency of neurologic disease or conditions in individuals with COVID-19 that set it apart. So that is very different from SARS-1 and MERS-CoV. There was some mild CNS involvement, but not very extensive, but there weren't that many cases, right? So it wasn't really studied that much. But neurologic symptoms are common in COVID-19. And so these could include in quote unquote benign symptoms, which are loss of sense of taste and smell, although that must be extraordinarily disorienting if you have this. In addition, individuals complain of dizziness and headaches. Again, stroke is a very serious problem. Seizures have been reported. And then again, inflammation of the brain. So this is encephalitis or meningitis. And what meningitis is simply immune cells have migrated to the meningeal sheath that encircles and protects the brain. In addition, loss of myelin, which is a lipid structure that protects axons. This has also been reported to occur in some individuals. All right, but a very, I think, disturbing and very important facet that's associated with COVID-19 are these individuals that have acquired the name long haulers. And so I captured the cover or the title of an article from STAT that appeared in August. And it really sums it all up. Long after the fire of a COVID-19 infection, mental and neurological effects can still smolder. All right, and I apologize for the text in this slide, but I think this is a very important component of this disease that we really need to understand better. So these novel coronavirus symptoms in some individuals can last weeks or even months. And again, these are referred to long haulers. And the one thing they have in common, they've recovered from COVID-19 and test negative for virus, primarily through nasal swabs, right? Doesn't mean the virus might not be persisting within the central nervous system, which is something I'm very intrigued about. Now, about 10% of COVID-19 patients can become these long haulers, all right? And interestingly, this can affect anyone, all right? Either young, old, healthy, no comorbidities, comorbidities. And it's been detected in patients who had severe forms of the disease that were hospitalized or those with very mild symptoms. And the symptoms are not simply restricted to neurologic conditions. There are a spectrum of symptoms that are distinct from CNS type disorders. But because this lecture is focusing on the neurologic aspects of COVID, I'm just listing here some of the symptoms of these long haulers within the context of neurologic disease. These can be headaches, which can be mild but can be debilitating in some people. The reoccurrence or the emergence of loss of taste and smell, even if these people did not originally have these symptoms. The most common complaint is brain fog, all right, where you just feel like you're in a fog, memory loss, and then, difficulty concentrating. Now, I just added this because there's a really nice paper that came out recently in bioRxives from Tony Weiss-Corey, who's a really sophisticated neurobiologist at Stanford University. And actually, Tony and I were postdocs together at Scripps. And what he reports in this study that in looking at gene transcription profiles in very specific neuron subsets in the brains of COVID-19 patients, he reports the expression of a constellation of gene subsets that have been associated with problems in cognition, and then very interestingly, schizophrenia, as well as depression. And I think the one thing I want to point out is that this number, about 10% of the COVID-19 patients, well, you might say, okay, 10%, that's not that many. But if you look worldwide, I think we're over 50 million people that are infected or have been infected with the virus, and most of them will survive. But what are the long-term consequences of this, particularly if the virus is able to persist within the CNS? And I think this is something that's really important to study. All right, so what are the questions that we, as well as others around UCI and as well as the world, are asking with regards to COVID-19 and neurologic disease? So what are the mechanisms by which this occur? And so how I've broken this down is, are there direct mechanisms? So in other words, can the virus infect and replicate within the CNS or cells specific to the CNS, which can either directly kill the cells or alter their function? Now we know from the literature that's emerging and that have studied neurological consequences of CNS infection, we can detect viral RNA in the cerebral spinal fluid, as well as in the CNS vasculature. In addition, there are some reports of virus in the parenchyma or deep CNS tissue. Is there an acute, if you have an acute CNS infection by the virus, does this result in enhanced or increased neural inflammation? And this results then in tissue damage of the brain. Alternatively, or collectively, can virus persist within the central nervous system? And does this result in chronic neuroinflammatory conditions? And here's where we can use infection with mouse coronavirus to inform us on some of these processes or some of these events, because mouse coronaviruses love to persist in the CNS. And as a result, there is chronic inflammatory responses and ensuing neurodegeneration. Alternatively, you can have indirect mechanisms by which infection with SARS-CoV-2 could induce neurologic disease. And what I mean by that is simply, in some patients, you have this acute inflammatory response in the lungs, all right? And this is mediated primarily by soluble or pro-inflammatory proteins called cytokines and chemokines. And these can be secreted from the site of infection, but they can enter the central nervous system and do quite a bit of damage and or alter neurologic function. And so these two mechanisms are not exclusive, all right? They could be combined or it could be separate and distinct, but I think these are things we need to figure out. Okay, so very quickly, before I get into some of the data I want to talk about, there are a number of neurotropic viruses or human neurotropic viruses that exist that are capable of infecting and replicating within the CNS. These include members of the RNA virus family, DNA viruses, as well as retroviruses. No doubt, I'm sure all of you have heard of some of these viruses like West Nile, polio, HIV, herpes, Zika. And again, we have here the three human coronaviruses or zoonotic coronaviruses that we've talked about. Mild involvement with MERS and SARS-1, but potentially a lot more involvement with SARS-CoV-2. And here in this cartoon, I just want to talk about four main cell types that we feel might be very important. One is the astrocyte. The other is the neuron. This is the neuron cell body, and these are long axons that project from the body of the cell that are utilized by the cell to transmit either electrical or chemical symptoms or signals that allow the cells to communicate with one another. You have oligodendrocytes. Oh, by the way, these axons are protected by myelin, all right? And so the loss of this myelin sheath can be devastating. And you probably, this occurs primarily in patients with multiple sclerosis, right? But it seems to also be happening in people with, some people that are infected with COVID. And then we have oligodendrocytes. These cells make the myelin sheath within the central nervous system. And then we have microglia. And I'm going to come back to microglia in just a minute. So what happens in the CNS following infection? The idea is very simple. You inhale SARS-CoV-2, and presumably, undoubtedly, it goes to the lung, and that's where it establishes a beachhead or a primary site of infection. But keep in mind, if you're inhaling the virus, particularly through the nose, it can go up and infect the olfactory bulb within the brain. And the olfactory bulb does a number of things, very important things, one of which is controls your sense of smell. And we know from studies in my laboratory and others that you can take mouse coronavirus, for example, and administer it intranasally to animals, and the virus loves to get into the brain and replicate within the olfactory bulb. So it's not entirely not possible. Now, I've had a very new, but I can tell this is going to be a great collaboration with Dr. Bill Young, who is a professor here in the Department of Pathology. He was kind enough to share some pathological features that occur in people that have passed away from COVID. Some of these, it doesn't look like much, but collectively it can be very damaging. One is swollen astrocytes. You see these large cells that are kind of fusing together. These are astrocytes that become enlarged or swollen. Alternatively, you can have what's called red neurons. And what this simply is a result of acute ischemia, all right? And these types of neurons can be distributed throughout the brain, and it can be very damaging in terms of neurological function. Now, more severe examples are provided by a paper that came out in 2020, and this is from Claudia Lucanetti's laboratory, who is a fantastic neuroscientist at the Mayo Clinic. And she had this beautiful paper showing that in an individual that passed away from COVID-19, extensive myelin loss in white matter tracts of the brain that was then associated with extensive axonal damage. And this can have devastating consequences for neurologic function in individuals. So what I'm trying to get across is the notion that the neuropathology is extensive. We're still learning quite a bit about it because we just need more and more samples in order to better understand this. All right, so what are we doing in my laboratory? And when I say my laboratory, I want to reemphasize the fact that the subsequent studies I'm going to be shown is a complete team effort, all right, from individuals around the country and more specifically here at UC Irvine. All right, and so the first question is, or the concept I want to get across is that we can use other viruses to model SARS-CoV-2 biology and pathogenesis. And the advantage of this, one of the key advantages of this is that experimental work can be performed in what's known as a BSL-2 or biosafety 2 laboratory, all right? And what that simply means is you don't have to put on the quote unquote spacesuit and go into a negative pressure room in order to do experiments with the virus. You can do experiments in a regular biosafety cabinet that's been certified in a laboratory. Here is one of my graduate students, Colin Pachow, who's working in our laboratory, working with the virus I'm going to introduce here. And this is, for better ways to describe it, it's a recombinant virus. The backbone of the virus, it's a VSV, which stands for vesicular stomatitis virus. But I don't want you to worry about that. What I want you to know is that this virus has been engineered to express the spike glycoprotein of COVID-19 or SARS-CoV-2 on its surface, all right? And so this virus was developed in the laboratory of Sean Whelan, who's chair of microbiology at WashU. And then Sean was kind enough to let us use this virus. And then subsequently, we've been working very carefully or closely with Dr. Bert Semler and Dr. Alex Buen. Dr. Semler is the former chair of micro and molecular genetics here in the School of Medicine, as well as he is the director of the Center of Virus Research. And he is a very eloquent virologist. And Bert and Alex have been very helpful for our laboratory in assisting us in growing the virus and sequencing the virus and what have you. But you can use this virus to do a number of important experiments for studying aspects that can be related to SARS-CoV-2 infection, including determining the number of cell types in the CNS that are susceptible to infection. And so the question that we first asked is, can we infect different human cell types, so neurons, astrocytes, or microglia, with this virus that has on its surface the spike protein of COVID-19, all right? We can then look for virus in the cells and measure their growth. So this work would not have been possible without a fantastic collaboration I have with Dr. Leslie Thompson and Dr. Matt Blurton-Jones, both of which are professors in neurobiology and behavior here at UC Irvine, and the wonderful support staff that they have in their laboratories. They provided us human neurons. So Charlie here was able to give us human neurons for our work, and these are just beautiful looking cells. Andrea was able to provide us with human astrocytes. Andrea and Charlie work in Dr. Thompson's laboratory. And then Christina too was able to provide us with human microglia, and Christina works in Dr. Blurton-Jones' laboratory. And so to cut to the chase, we infected these different cell types with our VSV SARS-2 spike virus. And what we found after 72 hours was very interesting. We could clearly see infected neurons. And how we could see this is because this particular virus has been engineered not only to express the SARS-2 spike glycoprotein, but also a reporter gene called GFP, or green fluorescent protein. And so you can see here clearly a number of different neurons that have been infected by the virus. And under high power, you can see that these cells are, well, they're really beautiful in my opinion. You can see the virus that's extending down these long processes shown here, extending far out. And so this work was done by Colin in the laboratory, as well as Hema Oliveira. They really worked extraordinarily hard to get this system up and running, and I'm truly indebted to their hard work. Again, we could look at the growth of the virus in these neurons, and you can see at 24, 48, and 72 hours, there's a natural increase in the number of infected cells. But what was interesting is that we could not detect infected cells in either microglia or astrocytes, arguing that, at least within this experimental context, neurons were the primary cells infected. In addition, I'll just tell you and not show you the data that we could block infection of neurons by this virus by blocking ACE2, which again is the human receptor that binds the spike glycoprotein. Okay, so those are experiments that we have ongoing with regards to working in the BSL-2, but we also have initiated experiments working in the BSL-3. So in other words, how do you do experiment with SARS-CoV-2? So one, it's much more involved. You have to work in a high containment biosafety lab 3. BSL-3 is used for research or diagnostic work that involves microbes that are primarily aerosolized, including SARS-CoV-2. And so I think there are several very important things I need to point out before I show you some of the data that we've acquired. One, we have 2 state-of-the-art BSL-3 laboratories on the UCI campus. And UCI is the designated national training center for BSL-3 work around the country. So in other words, people from all over the US come to UC Irvine to learn how to work in a BSL-3. And this has been made possible by Gary Landucci, who is truly outstanding, and how he's raised the visibility of BSL-3 work, not only on this campus, but nationally as well as internationally. And he deserves an enormous amount of credit for providing us investigators the facilities that we need to do this work. In addition, Eric Gwandike and Anjo Suba. Eric is the director of the high containment BSL-3 laboratories. Anju is the university safety officer. We have worked extraordinarily closely with Gary, Eric, and Anju because working in the BSL-3 requires extensive training. Anything we do requires a standard operating procedure that we work and edit and then rehearse before we go into the BSL-3. I also would be remiss not to mention Dr. Ilhelm Moussaoudi, who is a professor in molecular biology and biochemist. She is an extraordinarily sophisticated virologist and or immunologist who's doing some cutting edge work in COVID-19, looking at different patient populations. And then finally, and I will move on from this, but I just have to recognize these people because they're so important for our work. I've introduced Hema, who's a second-year student. There's also Susanna Furman, a second-year graduate student in my lab, Yuting Cheng, who is a fourth-year graduate student. And these are the true heroes of the work I'm going to show you. They are the people that are going in on a daily basis into the BSL-3, working with the animals, collecting tissues, infecting them, et cetera, et cetera. And none of this work would have been possible without their dedication to doing this. All right. So what we primarily do in the BSL-3 is infect mice with virus, all right? Specifically, we infect mice with SARS-CoV-2, but unfortunately, it's not that easy because normal mice they have ACE2, but their ACE2 is not compatible with SARS-CoV-2, all right? So they're resistant to infection for the most part. So we've had to acquire transgenic mice called K18 mice for reasons I'm not going to go into. But these animals have been engineered to express human ACE2. All right, so we acquired these animals from the Jackson laboratories, and then we use them for infection to look at different responses in the brain or lungs. And you can use these transgenic mice for a number of important experiments. One, to evaluate antivirals, as well as vaccine development, anti-inflammatory responses, as well as just simply to characterize responses that can occur with regards to defense and disease. And here I would like to sincerely acknowledge the efforts of Dr. Grant McGregor. Grant is a professor of developmental biology or developmental and cell biology here at UCI. He's also the director of the transgenic mouse facility. And then Dr. Shimako Kawuchi, who is really works very closely with Grant. All of the work that we've done with the K18 mice would not have been possible without their extraordinarily help in developing or allowing us to rapidly expand this colony of K18 mice. And so I'm truly indebted to them, as well as the Office of Research, for building the outstanding transgenic core facility that we have here. Okay, so the experimental goals, and I'm almost done, is that we want to infect these K18 mice with virus. How we do this, it's an intranasal route of administration. So we simply anesthetize the animals and you put a little drop of liquid containing virus on their nose and they inhale it and they're infected. But we wanted to establish that this was a good mode of infection and then determine if this allowed for infection of lungs and brains. Others have certainly reported this, but you really want to make sure you can do it yourself. And then in addition, we really wanted to evaluate the functional role of this cell I mentioned earlier, microglia, in both host defense and disease. So microglia are considered the immune cell of the brain. And we and others have shown that viral infection of the CNS does activate microglia. And this activation contributes to both host defense as well as disease by secreting these cytokines and chemokines that influence immune cell infiltration into the central nervous system. And so just quickly highlighting the importance of this, we published a paper earlier this year clearly showing that microglia impact host defense, disease, as well as repair in a model of mouse coronavirus infection of the CNS. These are the individuals that did the work that are shown here. Vishali was a former student in my lab. Dominic just defended his PhD. Colleen Stone was a former laboratory manager of mine. And then Kim Green is a great friend and collaborator I have here at UCI, who's an expert in microglia. So again, the question is, do microglia influence host defense or disease in response to SARS-CoV-2 infection? And so how we can address this is we can specifically ablate microglia in the brains of mice prior to infection with virus, and then evaluate disease progression, tissue damage, as well as viral burden is in the brains and lungs. And how we do this is it's great. And why is it great? I'll tell you why it's great. Because we can use a drug called Plex 5622, and this drug selectively depletes microglia upon administration to mice, all right? And the great thing about it is this drug is formulated in mouse chow. So all the mice have to do is eat the chow that contains the drug, and it gets rid of microglia. So this is actually what the Plex 5622 chow looks like. It actually is pink. The control chow is shown here in white. These are K18 mice, separate cohorts. We feed either Plex 5622 chow or control chow for 7 days to the animals. This will get rid of about 95% of the microglia in these animals. We then infect them with virus intranasally and collect brains and lungs 1 and 2 weeks post-infection. So this shows Hema, Susanna, and Yuting prior to going into the BSL-3. This is me. I actually do all the infections. I'm just an ardent believer. If I'm sending these students into the laboratory of the BSL-3 to do this work, I need to be in there with them to do it. And you can see it's quite the get up that we wear. Again, Kim Green is a collaborator for these studies. Kim is smiling because he is not dressed in the BSL-3 garb and because he's not going in. All right, so what happens when we do this? Well, again, these are preliminary data. We've acquired these over the past month and a half. It's been an extraordinary, long, involved experiments. But in the Plex 5622 treated animals, that is, they don't have microglia, we start seeing an increase in clinical disease severity that's associated with weight loss, all right? And so that means these mice are sick. And this argues the idea that Plex 56 mediated ablation of microglia increases disease onset as well as severity in the SARS-CoV-2 infected animals. And what's interesting here is now lung tissue that we acquired from these animals at day 8 and 9 post-infection. And this was work that we did in collaboration with Rob Edwards, the pathologist on campus. And then the staining for the virus was performed by Hema Oliveira. And what we're looking at is viral RNA. And what jumps out at us was animals on the control chow had much greater amounts of viral RNA in the lungs compared to those animals that were infected with SARS-CoV-2 but treated with Plex 5622. And you can actually quantitate this using quantitative PCR. And this is what Colin Patchow has done in the laboratory, very rapidly, I might say. And you can see there's a quantitative difference in the fold change compared to uninfected animals, the amount of viral RNA in the lungs of animals on controlled chow versus Plex 5622. Now, what's happening to the brains, all right? So here we're focusing on the area encircled in this sagittal section showing the mouse brain. This is the olfactory bulb, but this is the cerebral cortex. And mouse coronaviruses love the cerebral cortex, all right? Primarily because there's a lot of neurons. And so this is kind of a low power image just showing that there's a lot more virus in the cerebral cortex of animals treated with Plex 5622 compared to control chow, arguing that the Plex 5622 ablation of microglia increases viral burden in the brain. And indeed, you can look here and you can quantitate this. Again, this is work done by Colin, and there's a dramatic increase in the amount of viral RNA in the brains of Plex-treated mice versus control. And so we just got this data Monday. And again, it highlights the incredible collaboration we have with Rob Edwards, and then Hema did all the staining for virus. And this is odd to say, but these are just beautiful images, all right? And so this is in the cerebral cortex of Plex 5622 treated mice. And what you can clearly see here are neurons. We haven't technically immunophenotyped these cells, but we're in the process of doing this. But these are neurons that have been infected. And this is just a high power image of these cells here. And you can see these from the cell body, these long extensions extending that are infected, which raises some really fascinating questions about how the virus spreads in an infected cell. In addition, we see in the cerebral cortex these infected neurons, but what I'm pointing out here is what's known as meningeal inflammation. These are immune cells that are migrating into the meninges, similar to what's been reported in patients with COVID-19. In addition, this is perivascular cuffing or perivascular inflammation. This is the vasculature within the central nervous system, and these are the highways that the immune cells employ to traffic into the brain in response to infection. And you can see this is loaded with inflammatory cells that are then migrating into the parenchyma to help control infection. All right, so to put everything in perspective, and I'm concluding here, and again, thank you for your attention. Cultured human neurons express ACE2 and are susceptible to infection by virus expressing the CoV-2 spike glycoprotein. Importantly, we can block infection by blocking ACE2. Cultured human microglia and astrocytes are resistant to infection, at least under the experimental conditions that we've employed to date, although we are continuing these studies. Intranasal inoculation of human ACE2 transgenic mice, that is the K18 mice, with SARS-CoV-2 does result in infection of both the lungs and the brain. This is consistent with other reports. What we've determined is that neurons appear to be a primary target of infection by virus, but we need to look at later times post-infection to determine if other immune cell, or excuse me, CNS cell types are infected. Importantly, I believe this is a unique observation that we're working on, is that targeted ablation of microglia via Plex 5622 reduces the viral burden in the lungs, but increases the viral burden in the brain, arguing that microglia may be important in controlling SARS-CoV-2 replication and spread within the brain. So these are obviously areas that we're looking. All right, this is my last slide because I think it's important to discuss where are we now, all right? And so what I've talked about today is just a very small portion of some of the amazing work that's going on the campus of UCI, as Frank alluded to at the beginning of the talk. There are a number of outstanding laboratories that are contributing to our knowledge base on the basic biology of SARS-CoV-2 with regards to both the virology and immunology, clinical aspects associated with infection, the development of novel therapeutics that can be employed that can limit viral replication, and then finally vaccine development. And I would say the majority, if not all, of these ongoing studies were initially facilitated by funding of COVID pilot grants that were provided to us through the UCI Office of Research. And why this is important is because this provides seed funding to investigators to then subsequently apply for a number of larger grants to the National Institutes of Health, for example. And so I gratefully acknowledge the Kraft COVID Awards from the Office of Research. Finally, and this is my last slide, I have to acknowledge several people. Cynthia Manlapaz is my laboratory manager, and she technically is my boss. She makes my job exponentially easier. She runs the show beautifully. She's in charge of the ordering. She does everything, and it just seems she's graceful, and she works beautifully with everybody in the laboratory, and none of the work that I would have been able to do would have been possible without her. These four students that I've talked about extensively are truly amazing. I cannot say enough good things about them. They are amazing in what they've done with the work that we're doing. And Amber and Dominic are also amazing, but they're working on our mouse coronavirus projects. Rob Edwards, again, and the Experimental Tissue Resource Center that he directs. None of the images that I showed on human samples would have been possible. Again, I also need to acknowledge Bill and his contributions, Ilhelm and her contributions. She's my partner in crime in discussing experiments with COVID-19 and her wonderful support staff. Leslie and Matt and Kim for being there to help provide tissues and reagents that we acquire. I also have to acknowledge Dean LaFurla, who is a tremendous dean, and the support staff in the office of the BioSci. And for the sake of time, I do have to again acknowledge Gary, Eric, and Anju for allowing us to get into the BSL-3, Charlie, Andrea, and Christina for their help in getting us all these samples. Wonderful collaborators here that we'll be working with, Eric, Mike, and Craig. Stan Perlman, who helped us providing virus, Sean with also virus, and other collaborators. And I also would like to, again, acknowledge the funding for the laboratory. And Frank, thank you again for this opportunity. And I'd like to thank everybody for their attention.
Thank you. Tom, thank you so much. I mean, this was a tour de force, not only in terms of the way you presented all of the material, but just in terms of the amount of research progress that you have made. I mean, you have to remind everyone that essentially you started working on this March of 2020, and the progress is just impressive. So congratulations to you and your team. There, as you can imagine, a number of questions from many people, and so we'll try to be respectful of the time, but see how many we can get through. And I guess the most relevant question to ask right off the bat is, what are your thoughts about the Pfizer vaccine that was just recently announced?
Yeah, I think it's great. We need it. The 90% effectiveness, and simply what that means, and as probably most people know, it's effective in the occurrence of symptoms, right? And what we don't know to date, and Pfizer knows this, right, is whether this is really blocking infection, reducing symptoms. And there are a number of other important questions: is how effective is long-term memory that's evoked in response to the vaccine? But it's a great start, right? And I think we all, are beginning to breathe some sigh of relief. And there are a number of other really compelling vaccines in the pipeline as well. And so this is a great start. And so I'm very encouraged by this.
Can you maybe expand on what your thoughts are about immunity for SARS-CoV-2? I mean, if you go through this horrible disease, do you have to look forward to getting it again?
That's the question, right? That we all are worried about. I think, Frank, the interesting point is, believe it or not, we can learn quite a bit from those common coronaviruses, because we know that over time, your antibody levels wane, all right? So, about 5 to 6 years following infection, the antibodies to these relatively benign upper respiratory tract infections can go away, and you are susceptible to reinfection, all right? Now, whether this is going to happen with SARS-CoV-2, we don't know. A lot of people, social media has really grabbed onto this notion that antibody titers wane over time. But in the big picture, antibody titers do wane over time, all right? And that happens under most infections, all right? The main thing now is we just have to keep in mind that this disease, although it seems like it's been an eternity, it's been 8 months, right? And we have a lot more to learn about the immunology, the basic biology, but we'll get there. We're going to get there.
I mean, I think you know that my PhD was in virology, and I worked on vesicular stomatitis virus myself. And back in the 80s, people would talk about how fascinating coronaviruses were, but that there were no really relevant human
Yeah,
diseases that were impacted by them. And that has certainly changed.
yeah,
So I guess raises a question let me raise a question someone brought up, which was, how was the original SARS contained in 2003? And are there any lessons that we can learn from that?
yeah. Thank you for the question. It was contained primarily because it was a really crummy spreader, right? Because you had to be very sick and have symptoms in order to spread the virus. And once that was determined that family members and healthcare workers were the primary people exposed, you could easily quarantine infected individuals. And that's the same that's true with MERS-CoV. So effective quarantining, masks were apparent, obviously, just from the photo of time, but a lot of techniques that we're using now, right? But the problem is SARS-CoV-2 is much more insidious because you can be asymptomatic and very efficiently spread the virus.
Right. So a few of our professors have a very similar question. So Professor James Hicks and Robertson want to know if the commonly prescribed drugs that upregulate ACE2 receptors in the brain are possibly making people more susceptible to infection.
Yeah, Jim, great question. A lot of people interrogated that at the beginning, but it kind of tailed off. So if it had, you would have heard about this to great extent. So I don't think there's a really solid literature base in that right now. Now we can test that potentially in these animal studies, but I think I'm more interested in looking at anti-inflammatories and if that limits the spread of the virus or reduces the pathology. So that's an area that probably requires a little more investigation, but it's not prominent in the literature right now.
Okay, then if SARS-CoV-2 is able to enter the brain, are there any antiviral drugs that can penetrate the CNS and inhibit viral replication?
Wonderful question and extremely relevant because it's not clear if a vaccine will be able to evoke a memory or get virus in the brain. So developing drugs that are CNS penetrant is not trivial. And so remdesivir supposedly has good penetrance into the central nervous system, but I don't know if that's been tested to great extent in the animal models. And I think it's important to put this in the context of HIV because we have the cocktail of drugs that work great at reducing viral burden in the periphery, but a lot of people that are HIV infected can have virus in the brain and it is a reservoir. These drugs do not penetrate the CNS and these individuals some of them can develop what's known as HIV dementia. All right, so this is one of the areas that I'm very interested in investigating in my laboratory, particularly within the context of long haulers, right? So what if people that have these neurologic disease for years and years and years, is it due to a persistent viral infection? We don't know. And that really emphasizes the importance of characterizing drugs that get into the brain.
So another question relates to the findings of COVID with individuals with epilepsy and what's known about that.
It's probably a comorbidity. What we don't know is if there's greater CNS penetrance. Clearly, neurons are involved with epileptic seizures. And so are these seizures occurring or enhanced seizures occurring because of viral penetrance or infection of the CNS and neurons? To date, that's pretty much the extent I know of this. And it's certainly an area that requires greater investigation.
Okay, excellent. So maybe explain to us how lung inflammation can exacerbate disease in the brain. What's the connection there?
Right. So the easiest way to think about this is when all of us are infected by a virus or any type of microbial infection, one of the first things that happens is we acquire a fever, right? And that fever is your body's one of the first defense mechanisms because microbial pathogens don't like elevated temperatures. How we raise our temperature in response to infection is via cytokine secretion in the periphery. All right. So certain cytokines like interleukin 6 and TNF alpha could get into the brain and go to an area of the brain called the hypothalamus, which regulates your body's temperature, right? So it's almost like the rheostat. So this is an example how peripherally derived cytokines can influence factors in the brain. Now, in patients that have COVID or severe COVID infection, they often have cytokine storm that's elaborated initially in the lungs. These cytokines could then enter into the central nervous system and cause a number of different changes that can be either acute or potentially more permanent. So that's the notion right there of how this can happen.
Okay, Rick Robertson asked another question. Does the immune response depend on naive T cells? And those of us who are getting older don't have many naive T cells left.
Well, any type of a virgin infection, you need naive T cells that are bearing, cognate antigen receptors that can expand to the infection, right? And so certainly as you get older, the efficacy of your immune response wanes over time, right? We know that from vaccination trials and what have you. But that's why I think the priority for these first round of vaccines will be for the elderly. Like, this last week I was visiting my father who's 90 years old, right? And so, you want these people to be protected. So the older you get, the more effective your T cell and antibody responses are. In addition, your secretion of interferon in response to infection also diminishes as you get older, right? And that's truly one of the first responses that are required to control any type of virus. But that's also particularly true for SARS-CoV-2.
So let's make this the last question, considering the hour. And I guess it's the relationship between vaccine development and antiviral medications. And just what are your thoughts on that, the relationship between those two? Obviously, both are going to be critical in the response to COVID.
The big picture, you need a vaccine, right? I mean, if anything has proven over the past century is that vaccination is truly one of the greatest contributions of medicine has made to humanity. So we need a vaccine, but in the short time, there are antivirals can be very effective in limiting hospital stay or limiting even entry into the hospital, okay? And so a lot of investigators in the field right now are looking, including our laboratory, are looking at not only novel antivirals, but repurposing FDA-approved drugs that are being used for other disease indications that you'd be surprised that could be very effective in limiting viral spread in COVID patients. So, I can't say one is more, in my opinion, the vaccine is more important, but it doesn't diminish the importance of developing effective antivirals. And, we're doing both. Primarily, we're working on antivirals in my laboratory with some great collaborators in chemistry, because chemists at the end rule the world. So that's the way it goes.
All right, thank you, Tom. That's a good way to end. And unfortunately, we can't get to the many, many other questions that are in the chat. But I just want to remind everyone that the 2020 Dean's Report was largely devoted to the COVID-19 pandemic. So if you're interested in learning more about not only what Tom is doing, but what other faculty in the school are doing, please visit our website at bio.uci.edu, and you'll find a lot more information. You'll be able to download the Dean's Report and see what we're doing there, and hopefully have some of your questions answered. I also, Tom, want to just before we depart, again, thank you for this tremendous work that you're doing, for being such an outstanding colleague and such a friend to so many of us. And you pointed out the money that you got, the pilot award that you got from the Office of Research, and a significant amount of that also came from the School of Biological Sciences. And so I just want to make the point to the individuals in the audience that this is oftentimes where your contributions to the School of Biological Sciences, particularly to the Dean's Excellence Fund, allows us to support this kind of novel, innovative research. And I think it's because of these funds that we were able to disperse to you and to other researchers in the school that we've been able to make so much great progress, and not only on the research front, but also to help our students. So if there's anyone in the audience that's interested in helping or learning how you could become more closely affiliated with us and join our BioSci Ambassadors program, these are not programs that require huge donations, but just contact either Andy Denuzzo or Robin Martinelli. Their information is up on the screen right now. And you could see, I think, firsthand the direct impact that this kind of support has for all of society. So with that, again, Tom, thank you very much. And let me wish everyone a very safe and wonderful holiday season. And we'll see you for our next Dean's Distinguished Lecture in the winter quarter. Thank you all. Bye.
Good night.